Hi everyone, and today we would like to present our group presentation, and we are proposing a project for the product of ethical testing solution in advancing skin tissue engineering for cosmetic. And our group members are comprised of 5 members. They are Intan Balkis binti Muhammad Sattar S70787, Nur Sofia Camelia binti Jaafar S72212, Christina binti Jamaluddin S70834, Wan Nurul Nazifa binti Wan Zakaria S71916, and also Muhammad Shafiq Rizwan bin Ismail S70617. Basically, our objective of the proposal project is uh, to understand the description of why cells are called the building block of life, name the part of a light microscope, and also explain how to use the microscope. Basically, what is a cell? A cell is a living thing that may be one of the cells that comprise of a unicellular block of organism, or even or many cells that is known as the multicellular organism.
Let's start by addressing the core problem our project aims to solve, which is animal testing in the cosmetics industry. Animal testing is a practice that has been used for decades, but it is increasingly viewed as unethical and outdated. More than 100,000 animals, such as rabbits and guinea pigs, are used annually in cosmetics testing. These animals endure painful tests, often leading to severe suffering and death. Beyond ethical concerns, animal testing poses significant scientific challenges. Research shows that animal models are only about 60% reliable in predicting human reactions. This leads to product recalls, increased costs, and slower product development. Additionally, the rise of consumer awareness and demand for cruelty-free products has placed significant pressure on the cosmetics industry. Ethical considerations, combined with the growing popularity of certifications like Leaping Bunny and cruelty-free, highlight the urgent need for the companies to adopt alternative testing methods that align with consumer expectations and industry trends. Regulations banning animal testing are being implemented in many regions, such as the European Union and India, but companies face a critical challenge, which is to find reliable human-relevant alternatives to animal testing. This is where our project comes in. We propose the development of in vitro skin tissue models that replicate human skin properties. These models offer a cruelty-free, scientifically accurate and scalable solution for cosmetic safety testing. By addressing this issue, we can help the cosmetics industry transition to more ethical and sustainable practices while maintaining high product safety standards.
Animal tissue culture process. So this is the overview for animal tissue culture in skin engineering for cosmetic. Number 1, collection of skin samples. We got a source, collect samples of human skin usually from surgical waste such as skin leftover after dermatological treatments or plastic surgery. Since these tissues are not extracted especially for testing, they are regarded as ethical. To the concern, to guarantee the ethical sourcing of the samples, it is imperative to obtain the informed consent of the skin tissue donors. Next, isolation of primary cells. For the keratinocytes, these cells can be separated from the epidermis and make up the skin outermost layer. Fibroblast is located in the dermis, and these cells are essential for fostering skin regeneration and producing a supporting extracellular matrix. For other cells, you might also separate endothelial cells for vascularization or melanocytes for pigmentation, depending on how complicated the model is. Preparation of culture medium. Create a specific culture medium that resembles the skin cell's natural habitats. Essential nutrients, growth factors such as TGF, beta, and EGF, and cytokines needed to promote cell growth, differentiation, and maintenance should all be present in the medium. The medium for keratinocytes frequently consists of a mix of additives to encourage differentiation and basal mediums such as DMEM or keratinocytes SFM. A distinct formulation is needed for fibroblasts in order to promote their proliferation and the creation of a collagenous matrix. 3D skin tissue engineering. Fibroblast layering. Start by cultivating fibroblasts within a scaffold made of collagen for the cells to attach to, proliferate, and create collagen, which resembles the skin's dermal layer. This offers a supportive metric. Keratinocytes layering. The epidermis is created by seeding keratinocytes on top of the major fibroblast layer. The keratinocytes will multiply and develop into layers that mimic the skin's natural composition. Stratified skin formation. These keratinocytes will eventually create a stratum corneum, the outermost layer that acts as a barrier after building a multi-layer structure with a basal, spinous, and granulose layer. Cultivation in 3D. A 3D culture system in which the skin model is growing in an air-liquid interface is frequently used to replicate genuine skin. This helps to replicate the skin's natural conditions by exposing the skin model to air on one side, the surface, and immersing it in a nutrient-rich liquid on the others. To encourage differentiation, maturation, and the development of the skin barrier, the skin model is incubated in a humidified, regulated environment. Optimization for cosmetic testing. Barrier function. Make sure the skin models form a robust skin barrier, which is necessary for evaluating the efficacy and permeability of cosmetics. Relevance to testing. Evaluate the effects of different cosmetics on the engineered skin tissue, including toxicity, irritation, and absorption. Customization by include particular components such immune cells or melanocytes. The models can be altered to depict various skin disorders like acne, eczema, or aging skins, or reaction to various environmental factors like UV exposure. Ethical testing and validation. Product testing. The safety, absorption, and irritation of cosmetics can all be examined. This concept reduces the need for animal testing, which makes it more morally acceptable and applicable to human skin. Alternative toxicity testing. The methodology can be applied to various toxicological evaluations, such as determining the potential for irritability of chemicals and medications in addition to cosmetic testing. Endpoint assessments. To evaluate the quality and functionality of the created skin tissue, conduct viability tests, gene expression profiling, and histological examination. Examine the skin models, barrier function, and tight junction development, including tight junction proteins like occludin, to make sure it closely resembles actual human skin. Acclimatization and use in product development. Long-term testing of cosmetics for skin restoration, anti-aging, and other therapeutic uses can also be conducted using the skin model. The model can also be modified to investigate how pollution or UV rays affect the skin and evaluate possible cosmetic made to mitigate these environmental impacts.
The development of in vitro skin tissue model for ethical cosmetic testing has significant potential for commercial impact. These models are not only aligned with the growing ethical and regulatory demand, but also present numerous market opportunities across industries, particularly in the cosmetic industry. So here is a detailed analysis of the commercial impact. In many regions, including the India and parts of the US, animal testing for cosmetic is banned. So in vitro model provide a compliant alternative. As a result, this brand can continue product development while meeting the ethical standards. So it is compliant with regulatory requirements. Next is market positioning. Ethical and cruelty-free testing method resonate with environmentally conscious consumer. So the company can market the product as a cruelty-free, vegan, or ethically tested, and attracting a larger consumer base. Besides, faster testing cycle compared to animal model accelerate time to market for new product. And it is reduced costs associated with animal testing facilities and long regulatory approval timeline. That is the cost-effective innovation for this development. Faster testing cycle compared to animal model accelerate time to market for a new product. It is reduced costs associated with animal testing facilities and long regulatory approval timeline, as in the cost-effective innovation. For a global expansion, it enables the company to enter market with stringent regulation against the animal testing. Beyond cosmetic, in vitro models have application in biotechnology and pharmaceuticals, including drug testing and personalized medicine. So this broader application create opportunities for partnerships, licensing, and revenue generation. Companies adopting this technology can position themselves as a leader in ethical innovation, unlocking access to premium market and fostering the customer loyalty. Financially, transitioning to in vitro model reduces costs associated with animal care while opening new revenue streams through high-demand ethical product lines. With the global alternative testing market projected to grow substantially, these models are poised to drive sustainable growth and reshape the testing practices across industries.
Hello, my name is Kisina binti Jamaluddin. I will talk about call of action. Call of action. Join the movement for ethical beauty. End animal cruelty. Demand an immediate halt to animal testing in cosmetics. Advocate for a world where no creatures suffer for beauty. Embrace innovation. Support cutting-edge alternatives that ensure safety without harm. Collaborate for change. Partner with organizations, brands, and researchers dedicated to cruelty-free standards to create a unified impact. Champion sustainability. Encourage brands to adopt eco-friendly testing methods that protect both animals and the environment. Commit to compensation. Choose and promote brands that align with cruelty-free and ethical beauty practice. Make informed purchasing decisions to drive change. End animal cruelty. Animal testing is outdated, inhuman, and unnecessary, causing millions of animals to suffer painful experiments. For cosmetic safety, demand global policy change and hold companies accountable for ending animal testing. Sign petition, support legislative effort, and boycott brands that engage in such practice. Next, embrace innovation. Modern science has provided us with incredible tools that eliminate the need for animal testing, such as in vitro testing, organ on chips, and computer model and AI. We can ensure products are safe without causing harm. Encourage brands to adopt this innovative testing method. Collaborate for change. True change happens through collaboration. Support partnership between brands, cruelty-free certification organizations, and research institutions to accelerate the transition to ethical practice. Work with others to spread awareness, fund research, and amplify campaigns for cruelty-free beauty standards. Next, champion sustainability. Ethical beauty complements sustainability. Animal testing frequently requires extensive resource usage, such as water, energy, and toxic chemical disposal. Sustainable options minimize waste, carbon footprint, and environmental impact. Support brands that use ecological responsible testing and packaging. Together, we can safeguard animals and the environment. Lastly, commit to compensation. Every consumer has the power to drive change. Choose cosmetics certified cruelty-free by trusted organizations like Leaping Bunny or PETA. Educate yourself about the ethics of your favorite brand. Share your knowledge with friends and family to create a ripple effect of compassionate choice. Remember, every dollar spent on cruelty-free product is a vote for a kinder future.